Good morning and warm welcome to all the participants. Today we have with us Dr. Anupama T.K. Ma'am is scientist ICAR-Central Institute of Fisheries Technologies, Veraval Research Center. And the topic chosen for today's deliberation, it's microbial toxins in seafoods. Now I asked Dr. Faisal to give a formal introduction of Dr. Anupama. Dr. Faisal, if you are able to hear me, please start with the formal introduction, and then Dr. Anupama can start.
Okay, thank you. Good afternoon to all the participants. Let me introduce myself first. I am Dr. Faisal, working as an assistant professor in the Division of Post-Harvest Technology. In today's session, we are with us Dr. Anupama T.K, who holds doctorate in post-harvest technology and presently working as an ARS scientist, having 8 years of professional experience from CIFT, Central Institute of Fisheries Technology, ICAR. Her scientific research is related to quality and safety of fish and fishery products, particularly microbial safety, working on microbial toxins, risk assessment of pathogenic bacteria, and predictive modeling. Over to you, ma'am. Hello.
Yeah, hello.
Hello. Thank you, Dr. Faisal.
Yes.
Please start.
Yeah. Very good afternoon. Good afternoon to all. And today, my topic for this presentation is microbial toxins in seafood. Since it is a very vast topic, I am restricting to only to bacterial toxins of public health significance. So, as the introduction, according to the FAO 2020, world fish production has increased to around 179 million tons in 2018, and nutritionally, the fish account about 17% of total animal protein, and 7% of all proteins consumed globally. And the world per capita consumption of fish is increased from 9 kg in 1961 to 20.5 kg in 2018. About 88% of global fish production are for direct human consumption. Of that, 44% of them are live, fresh, or chilled status, and rest of them is production. Rest of the production is processed with 35% frozen, 11% in prepared and preserved forms, and 10% of dried, salted, smoked, or otherwise cured. And of course, seafood is the most traded commodity in the world, and nearly 75% of volume of seafood in international trade is imported by the developing nations, and 50% of that is exported by the developing nations. So therefore, the food safety issues concerned with the seafood is no more a local or restricted to a particular geographical location, and it has an acquired global dimension. And according to the surveillance report of US Center for Disease Control and Prevention, and fish was a food category, and it is most commonly implicated in foodborne outbreaks. And according to the report of CDC, there are about 31 major foodborne pathogens are reported from the fish and fishery products. And in US, it was found in 10 years of there are about 937 foodborne outbreaks with fish, resulting in 5,011 illness and 364 hospitalization and 4 deaths. Percentage increased from 2011 to 2008, and approximately 7% of total annual food outbreaks is consumed to be associated with fish. And that is, in every year, there is about 6 to 8% of total confirmed foodborne illness is contributed only from the fish. And it is more higher than the other products like meat and chicken, which is about 1.6% and to 2.3%. And most illness is caused by the norovirus or Salmonella, Clostridium, perfringens, Campylobacter species, and other bacteria. So coming to the microbial toxins in seafood, the present topic of the day, and there are the two most common types of foodborne illness in human are intoxication and infection. And foodborne infections are caused by swallowing these live pathogens that grow within the body, usually in the intestinal tract. And intoxication is a condition caused by swallowing preformed toxins, that is, toxin produced by microorganisms in the food before it is eaten. And other type of toxin is also from the foodborne. It is called as toxico-infections, and it is also it is also known as the toxin-mediated infections. That is ingestion of the pathogens, and they are causing toxins either in the small intestine or large intestine. And these are the examples for intoxication and toxo-infections and infective infections. And coming to the foodborne bacteria that can cause intoxication in fish is Bacillus cereus, Clostridium botulinum, Clostridium perfringens, and Staphylococcus aureus. And these bacteria may produce even single or even multiple toxins are also produced. And this toxin production in food is influenced by the extrinsic and intrinsic properties in the food, cell density, growth phase, cell stress, and injury. And before going to the bacterial toxins, there are two types of bacterial toxins are there. And we'll come to what is bacterial toxin. And it is a macromolecule, mainly of protein origin, which can cause toxic damage in a specific organ of the host. And toxins, they are divided into two, endotoxins and exotoxins. Endotoxins are otherwise called as lipopolysaccharides. It is found in the outer membrane, or it is caused by the gram-negative bacteria. And it is considered as the most important antigen of the bacteria, and they are released into the medium after different process, or when the cell membrane get disintegrate, such as during the lysis or other cell division. This endotoxin is released into the medium, and they are capable of causing the endotoxic shock and tissue damage. Exotoxin, that is of a protein origin, and it is produced or later released to the medium by the microorganisms. And when other type of toxin is called as enterotoxin, which that is exotoxin, which causes the gastrointestinal diseases. That particular toxin is referred to as enterotoxins. So coming to the toxins, the Bacillus cereus enterotoxin. The Bacillus cereus bacteria are gram-positive, motile, facultative, anaerobic, ubiquitous bacteria characterized by their ability to form resistant spore coats. And there are about 48 non-species in the genus Bacillus, but only the Bacillus cereus is associated with the foodborne outbreak in fish. And they are mesophilic bacteria, and that produce heat-resistant endospores and grow between 10 degrees Celsius to 50 degrees Celsius with an optimum temperature of 35 degrees Celsius and 40 degrees Celsius. Food poisoning due to this organism occurs when the food are prepared or held without adequate refrigeration for several hours. And the food poisoning is caused by the ingesting these heat-stable enterotoxins. And since they are also known for psychrophilic bacteria, so this bacteria can capable of producing the food poisoning when there is not giving proper chilling after the pasteurizing process. And this bacteria causes two types of gastrointestinal disease, the diarrheal and the emetic syndromes, and which is caused by various types of toxins. And the emetic syndrome is caused by the emetic toxin celluride produced by the bacteria during the growth phase in the food. That is already it is a preformed toxin. And the other type is called as a diarrheal syndrome, and it is caused by the diarrheal toxins during the growth of bacteria in the small intestine. And the toxins which are involved in the diarrheal syndrome are hemolysin BL, a non-hemolytic enterotoxin NHE, and cytotoxin. And the symptoms for diarrheal syndrome toxin, it is symptoms is associated with onset of watery diarrhea, abdominal cramps, and pain occurs 16 to 15 hours after the consumption of this contaminated food, and it is mostly accompanied with diarrhea, and there is no vomiting. But in case of emetic type of food poisoning, it is characterized by the nausea and vomiting, and occasionally only the abdominal cramps and diarrhea may occur. And its intoxication or infection, intoxication dose is diarrheal syndrome. It is ingestion of more than Bacillus cereus cells of 10⁵ CFU in the food. That is the consumption of about 10⁵ CFU per gram. And emetic syndrome is consumption of about 10 microgram per kg of toxin in food. And diarrheal syndrome is caused by the toxins. The toxins which is caused by the diarrheal syndrome is hemolysin, HBl, and non-hemolytic enterotoxin, which causes hemolysis and cytolysis. And emetic syndrome, it causes the emesis by the action of a nerve vagus of the brain center. And toxic stability, diarrheal syndrome, it is toxic at 56 degrees Celsius at 5 minutes. And emetic syndrome, it is even toxic at 121 degrees Celsius for 90 minutes. And the control measures is the proper hygiene and appropriate temperature control, optimization of heat process and temperature control to prevent spore germination. And quick chilling methods to cool the food below 7 degrees Celsius within 4 hours preparation in case of the retorting. Now, coming to the Clostridium botulinum. Clostridium botulinum is a dangerous food poisoning organism, and it produces a very deadly exotoxin, which is called as neurotoxin when it grows in food. The food poison is known as botulism, and it is an anaerobic gram-positive spore-forming rod-shaped bacteria, and the spores are very heat-resistant, and 7 different toxins are known to be exist from A, B, C, D, E, F, G, and it causes nausea, vomiting, fatigue, headache, paralysis, difficult to talk, double vision, and sound in the ear are the usual symptoms. And symptoms develop within 18 to 36 hours of consuming infected food, and death also occurs due to respiratory failure. And mortality rate is very high, in about 10 to 50%. The organism is found throughout the environment and found in the intestinal tract of fish, gills, viscera of fish, and gills and viscera of crabs and shellfish. Survive in normal cooking temperature, and it is also grows in vacuum packed and modified atmospheric temperature. And it is a botulism is a problem in canned foods if it is improperly sterilized. And smoked, salted, and fermented fish are also identified as a cause of botulism. Clostridium botulism type E is the most common type in seafood, and it produces of a major, and its products is of major concern because it grows at a very low temperature, around 3.3 degrees Celsius, and produces a noticeable level of spoilage also. And 7 toxin types are responsible for these botulinum symptoms. And botulinum toxin type A, which is type A, it is a neurotoxin which has high fatality. It is about 1,000 times more toxic than tetanus toxin. And A, B, E, and F are mainly involved in botulism in human, while type C and D are involved in animals. And this botulism, Clostridium botulinum bacteria, is divided into 2 groups, group 1 and group 2, based on the proteolytic activity. And group 1 is proteolytic Clostridium botulinum, which is producing botulinum neurotoxin type A, B, and F. And Clostridium group 2, it is non-proteolytic Clostridium botulinum, and it is responsible for most cases of foodborne botulism. And for Clostridium botulinum, group 1 forms a very high heat resistant spores and is a concern in the safe production of canned products. And the botulinum cook, it is mainly concerned for the Clostridium botulinum group 1, and given to the cans with a low content of acid is designated to inactivate them. And Clostridium group 2 is able to grow and form neurotoxin at a refrigerated temperature as low as 3 degrees Celsius, and is a concern in minimally processed refrigerated food. And types of intoxications. There are three types of intoxications. One is foodborne botulism, wound botulism, and infant botulism. Foodborne botulism is usually spread by consuming food contaminated with a botulinum toxin or spores, and it causes a severe neuroparalytic disease. And wound botulism occurs when the spores of Clostridium botulinum get into an open wound and able to reproduce, and it usually rarely happens. And infant botulism occurs when infant ingests Clostridium botulinum spores, and the spores get deposited in the gut and later produce toxin, and this is also considered as a toxico-infection. And it's this neurotoxin, it is consist of a heavy and a light chain and linked with a disulfide, and its activity only where it depends upon the presence of zinc. It is zinc-dependent protease toxin, and its biological effect is it inhibits the presynaptic release of acetylcholine from peripheral chlorogenic neurons, resulting in the flaccid paralysis. Intoxication dose is 1 microgram per kg orally for a 70 kg man, and 0.09 to 0.15 microgram intravenously or intramuscularly, 0.7 to 0.9 microgram inhaled. And toxin stability is, it is stable up to 80 degrees Celsius for 10 minutes and a pH of 2.7 to 10.2. And protease, it is also stable to enzymes like in proteases, and it's not stable, it is sensitive, but proteolytic activity is needed for the activation of the toxin. And control measures are proper processing under hygienic conditions, and adequate retort are suggested as a remedy. Next is a Clostridium perfringens. Clostridium perfringens is a gram-positive, pleomorphic, non-motile, spore-forming, rod-shaped anaerobic bacteria. And the food poisoning caused by the Clostridium perfringens may occur when food such as meat or fish are cooked and held without maintaining adequate heat or refrigeration before serving. And this illness is self-limiting, and incubation period is 8 to 15 hours and duration of 12 to 24 hours. The symptoms, which includes intense abdominal cramps, gas, and diarrhea, and it is also called as the enterotoxin, and it is produced during the sporulation of organism in the intestine. So it is also classified as a toxico infection. Clostridium perfringens are estimated to be second most common bacterial disease of foodborne illness in the developed countries. As in US and all, it is causing 1 million illness each year. And Clostridium perfringens strains are classified into 7 types: A, B, C, D, E, F, G. And Clostridium perfringens strains, so in the 7 strains, type F is more relevant for foodborne toxico infections, and this species can produce about 17 different types of toxins, and 4 of these are alpha, beta, epsilon, and iota, which are responsible for the tissue lesions and host death, and considered as a major toxins. In alpha toxins, it's caused by the 8 strains of Clostridium perfringens, which cause gas gangrene and also hemolysis in the infected species. And beta toxin, and it is found in type B and caused by the type B and type C strains, and it is causes necrosis by way of increased blood pressure, and it is brought on presence of catecholamine. Epsilon toxin, which is produced by the type B and type D strains, and it is a very dangerous toxin, and it can create pores in the tissue, which can result in leaked potassium ions and fluid leakage. And iota toxin, the iota toxin is produced solemnly by type E strain, which is cause tissue death in the infected individuals. And intoxication or infection dose is about 10⁶ to 10⁷ CFE per gram. And the toxin production is produced in the small intestine of the host. And toxin stability is, it is stable up to 60 degrees Celsius for 5 minutes, and pH is 5 to 10, and proteases, during the proteases, it increases its activity. And control measures are prevention from cross-contamination of cooked food, cleaning and sanitizing of food contact surfaces after being used for raw materials, and it's an effective way to control. Now coming to the Staphylococcus aureus. Staphylococcus aureus is a gram-positive, non-motile, facultative, anaerobic, spherical, non-spore forming cocktail. And this habitant and source of contamination, the primary habitant of Staphylococcus aureus is man, and this organism is present in the sweat, ear gum, ears, throat, ulcers, boils, and nasal cavity. And it is known that 50% of human beings are carriers of this organism. And the fish which is caught from the open seas doesn't contain this bacteria, and it is contamination occur during handling. So the presence of this in the food indicates the lapse of personal hygiene. Few Staphylococci per gram of food is harmful, but the food poisoning outbreak occurs when the product is handled carelessly during processing. And this organism can multiply vigorously and produce toxin at a temperature near about room temperatures. The toxin once formed will not be destroyed even at 100 degrees Celsius, even though the organisms are killed at this temperature. And it is evident that once a sufficient quantity of toxin is formed before its consumption, food poisoning can follow, even the food material is cooked. Food most likely involved in Staphylococcus food poisoning are cooked and processed and having low number of competing microorganisms. And this is also considered as a most drought-resistant pathogenic bacteria and cannot compete with the general microbial flora. And all the strains of Staphylococcus aureus are not able to produce the enterotoxin. Only some strains, only some of the strains can have the capability of having this staphylococcal enterotoxin genes, and such strains are called as enterotoxigenic strains. The enterotoxin strains, staphylococcal enterotoxin function, it is not only causes a gastrointestinal diseases, but also they are also called as super antigens. That is, they can activate the T cells, which results the release of inflammatory cytokines. Until date, there are about 26 staphylococcus enterotoxins. In that, 5 are considered as classic enterotoxin, and which is they are divided into 2 types: classic enterotoxin and newly identified enterotoxigenic like proteins. Classic enterotoxin is A, B, C, D, A to E, and others are considered as a newly identified enterotoxin like proteins. And most of the, that is 90% of food poisoning is caused by this classical enterotoxins, and in that staphylococcal enterotoxin A causes 80% of the food poisoning, and staphylococcal enterotoxin B is considered as a bioweapon, and they can directly affect the intestinal epithelium and vagus nerve, which cause stimulation in the emetic center of the brain. And they are usually these staphylococcal enterotoxin gene are carried, encoded in different enterotoxins, and are disseminated in the various mobile genetic elements such as these prophages, plasmids, and chromosomal pathogenicity islands. And this is very difficult because they can cause additional risk in the staphylococcal food poisoning, food intoxication because of the horizontal gene transfer. And it is well known that these strains carry more than one enterotoxins encoding gene. This is one of the study which is carried out here, and it was found that these strains, there are about all the strains, most of the strains are carrying these genes, and the multiple genes like are also found in the Staphylococcus aureus, which is isolated. And these are the some of the gel picture which shows the presence of their genes, which isolates. And the disease symptoms are nausea, vomiting, abdominal pain, diarrhea, subnormal blood pressure are the usual symptoms. It may appear within 1 to 6 hours of consumption of contaminated food, and complete cure is possible within 48 hours. Intoxication or infection dose, it is 100 nanogram of ingested toxin or 0.05 nanogram per ml of food is enough for can cause the poisoning, and its stability is the enterotoxin A is stable even at 80 degrees Celsius for 3 minutes, and or 1 minute at 100 degrees Celsius, and B is 87 minutes at 99 degrees Celsius, and it can stable in a wide range of pH, so it and resistant to gastric pH. And for the enzyme activity, it is resistant to the proteases. Control measure is adequate control over the health and hygiene of fish handlers, and refrigeration of the material during handling and processing. Minimize time temperature abuse of seafood, especially after cooking. Now coming to the pathogenic E. coli, and the E. coli gram-negative rod-shaped non-spore-forming facultative anaerobic bacteria, and this bacteria that is commonly found in the gut of the human and warm-blooded animals. And most of the E. coli are harmless. Around 700 studies found that 700 serotypes are found as harmless, and other pathogenic E. coli which are classified into 6 categories. It is called diarrhea E. coli, which include enterotoxigenic E. coli, enteropathogenic E. coli, enteroinvasive E. coli, enterohemorrhagic E. coli, Shigella toxin producing E. coli, and enteroaggravative E. coli, and diffusely adherent E. coli. And pathogenic strains of E. coli are transmitted to seafood through sewage pollution of the coastal environment or by contamination after harvest, and also when they by the use of the ice which are used for the preservation, or also from the utensils which are contaminated with E. coli. And others are improperly cleaned. Other source of contamination is improperly cleaned bottles and containers used on onboard trawlers, and also cause the source of contamination. And among these, the Shigella toxin producing E. coli are most isolated, and it is the most toxic bacteria, and it causes severe foodborne diseases. It is transmitted to human primarily through consumption of contaminated foods. STEC producing toxins, which is known as Shigella toxins, because of their similarity to the toxins produced by the Shigella dysentery. And these can produce two types of toxins, which is called as STX1 and STX2. And STX1 is more or less identical to the toxin of Shigella dysentery, and STX2, which have about a characteristic similarity of STX1, and they can grow in a range from 7 degrees Celsius to 50 degrees Celsius with an optimum temperature of 37 degrees Celsius. And they can grow in acidic foods down to the pH of 4.4, and food with a minimum water activity of 0.95. This is the one of the study carried out here, and it was found that around 22.5% of the isolates having the showed the presence of these XTEC genes, that is STX1 and XTX2, and multiple toxins are also present in the E. coli isolates, which are isolated from here. And this Shigella toxin in E. coli is destroyed by thorough cooking of food until all parts reaches a temperature of 70 degrees Celsius or higher. And E. coli O157H7 is the most important STEC serotype in relation to public health. However, other serotypes have frequently been involved in sporadic cases and outbreaks. And symptoms are abdominal cramps and diarrhea that may in some cases progress to bloody diarrhea, that is called as hemorrhagic colitis. Fever and vomiting also occurs. Incubation period is about 3 to 8 days, and mostly the patient will recover within 10 days. But in some cases, the infection may lead to life-threatening diseases such as hemolytic uremic syndrome. This hemolytic HUS, hemolytic uremic syndrome, is characterized by adequate renal failure, hemolytic anemia, and thrombocytopenia. That is the low blood platelets. And the sources and the transmission, E. coli O157 is transmitted to a human primarily through consumption of contaminated foods such as raw or undercooked meat products, and fecal contamination of water and other foods, as well as a cross-contamination during food preparation. Person-to-person contact is another mode of transmission through oral fecal route, and it will also act as an asymptotic carrier, which is the individuals which show no clinical sign of disease but are capable of infecting others. The control measures are the only effective method is the bactericidal treatment such as heating, that is cooking or pasteurization or irradiation, and also following the basic good food hygienic practices. Now coming to Shigella. Shigella is not indigenous on food. The disease caused by Shigella is generally known as shigellosis, transmitted through food or water contaminated with human excreta, and is gram-negative, facultative, anaerobic, non-sporulating, non-motile rod-shaped bacteria. And man is the only non-natural host for Shigella. And transmission of this organism from one individual to another is by following through contaminated food, water, ice, etc., or through contaminated contact surface, through flies, and through food handlers who are carriers of this organism. And it is naturally found in the intestinal tract of humans, and this organism passes the acid barrier of the intestine, multiply in the gut, and produce ulceration of large intestine followed by dysentery. And Shigella dysentery causes the most severe illness, and where symptoms are abdominal pain, fever, watery diarrhea, blood and mucus. And Shigella dysentery is an heat labile exotoxin, and it is this toxin is similar to the E. coli toxin, that is EHEC toxin, and it can cause enterotoxic, cytotoxic, and neurotoxic effects. Its mode of action is it inhibits the protein synthesis and causing the death of susceptible cells. And this is a Shigella toxin. It consists of about 2 subunits, that is A and B. And this is and a B subunit which attaches the colonic epithelial cells, and A subunit enter into the cell by endocytosis, and it inactivates the ribosomal function and thereby depurinate or disintegrate the RNA, and thereby it inhibits the protein synthesis. Thereby it infects the human cell. And preventive measures is strict maintenance of workers, personal hygiene, and they have to follow the good sanitary and handling practices, avoid time or temperature abuse, and identify the and avoid carriers, and prevent the entry of flies to the food processing halls. Next is the Vibrio cholera. Vibrio cholera are gram-negative, comma-shaped and aerobic non-spore-forming motile rods, and the disease which is caused by the cholera, it is the causative agent of this disease is cholera, and the cholera there is consist of 2 biovars, which is called as classical non-hemolytic and EI-TOR hemolytic one. EI-TOR vibrios are generally more infectious than classical Vibrio cholera, and it can survive longer in the environment. And there are 2 types of serotypes, Vibrio O1 and non-O1. In Vibrio O1 is Ogawa, Inaba, and Hikojima, and newly added serotype is called as Vibrio O139. And which is a Vibrio, even though it is a non-O1, the O1 Vibrio with cholera, which shows the similar characteristics also, but they doesn't have this O1, but the non-O1 doesn't have the O1 somatic and antigens. So they are classified into this. Non-O1 cholera was classified into non-agglutinable vibrios or non-cholera vibrios. And this non-O1 Vibrio cholera used to produce mild diarrhea. Habitant is the only natural habitant of Vibrio cholera is man, and the source of contamination is the person can get cholera by drinking water or eating food which is contaminated with the cholera bacterium, and it is occasionally spread through eating raw or undercooked shellfish that are naturally contaminated. And symptoms includes nausea, vomiting, and profuse diarrhea, and after an incubation of 1 to 4 days. And the stool resembles the rice water, and it contains, and the stool contains a mucus or epithelial cells and large number of vibrios. And their mode of action is the toxin which is caused by the cholera is called as a cholera toxin, and it is also called as a enterotoxin because they are capable of causing gastrointestinal diseases, and it activates inside the cell wall. Once the cholera toxin enters, it increases the adenyl cyclase activity and thereby increase the levels of intracellular cyclic adenosine monophosphate and promoting the fluid, and thereby it secretes the electrolytes into the intestinal epithelium and thereby causing the diarrhea. And it is a very potent exotoxin. And this toxin, the presence of this toxin is usually identified by the presence of the gene CTX-AB gene. And Vibrio cholera, no one also used to have this CTX gene, but it is very rarely expressed. And the preventive measures for this is proper disinfection of contact surface and avoid cross-contamination of cooked products and strict personal hygiene of seafood or food handlers. Coming to the Vibrio parahemolyticus, it is a marine pathogen present in marine and brackish water environment, and it can cause food poisoning when it is consumed in large numbers, about more than 10⁵. And it is usually this type of is found in the people who are habit of eating uncooked seafood, that is like Japan. Then the onset of symptoms is within 24 hours of eating this infected food. And Vibrio parahemolyticus, it is a gram-negative, non-sporulating, halophilic, motile oxidase positive or rod-shaped bacteria. And the symptoms are abdominal, usually after the symptoms includes the abdominal pain, vomiting, diarrhea, and fever are the usual symptoms. And in the case of the Vibrio cholera, there are two serotypes, oven and non-oven. Here, the Vibrio parahemolyticus, they are also considered with two types are there, Kanagawa positive and Kanagawa negative. The Kanagawa positive is considered as a pathogenic, and negative is a non-pathogenic. And a newly found O3K6 serotype is also added into the Kanagawa positive bacteria. And the isolates which are from the clinical sources are Kanagawa positive, that is, they can ability to produce the thermostable direct hemolysin, that is a TDH gene. And whereas the isolates which are found from the marine sources are Kanagawa negative. And along with the TDH, the other gene which are present in the Kanagawa positive is a TRH, that is thermostable related hemolysin, which have almost it shares about 60% similar to the TDH, and these both are associated with both these the strains which having these two are causing the gastroenteritis. And many clinical strains of these parahemolyticus can produce both TDH and TRH. And for the preventive measures are icing the materials immediately after the catch, and improvement of hygiene are the remedial measures, and cooking, thorough cooking of the seafood, and the preventing the cross-contamination of food after cooking with the raw material. And control of time and temperature abuse is also an important preventive measure. And Uptitil is the bacteria which are causing this intoxication, or they are classified into as a toxic infection. And this Vibrio vulnificus, which are classified in the classified as a foodborne infectious bacteria. And this Vibrio vulnificus, and it is an emerging pathogen which is phenotypically similar to Vibrio parahemolyticus. And the etiological agent is for three syndromes: primary septicemia, or skin infections through the acute diarrhea, and entry into the human by two portals, ingestion of raw seafoods or exposure of skin lesions. And its mortality is also very high. It is around 60%, and it is also a normal bacterial flora of estuarine and marine water. And it is Vibrio vulnicus is a gram-negative, halophilic, lactose positive, and rod-shaped bacteria. All the strains which are found as pathogenic, and the infection dose is not known. And the infection is associated with the consumption of raw seafood, particularly these oysters and clams. And for the disinfection, 10-minute treatment at 50 degrees Celsius is adequate to reduce Vibrio vulnificus to a non-detectable level, and icing is very effective to reduce the load of this organism. And this organism is closely associated with the oyster tissues and can be easily removed by using the UV assisted depuration systems. And now effective means are commercially exist for the elimination of this health hazard in oyster in the food. And so it is advised to avoid this raw seafood completely. Now coming to Salmonella. Salmonella, it is also classified as a foodborne infectious, and it is also known as the enteric organism, that is, they can producing the enteric fever and foodborne gastroenteritis. And till date, there are about more than 2,600 serotypes of these organisms are known exist at present, and more are added to the list every year. And the food poisoning, it is caused due to the salmonella, is known as salmonellosis. The infant, elderly, and undernourished are susceptible to this disease. Even the presence of a single cell of salmonella can cause the food poisoning or food salmonellosis. And it is a non-spore forming, motile, mostly motile and facultative anaerobic gram-negative rods. And its a primary habitat is the intestinal tract of the infected humans or animals, or in the warm-blooded animals like insects or lizards. And it is introduced in fish and fishery product through the contact with the contaminated water while during the handling. It is without proper control and other inappropriate fish breeding, processing, or storage practices. And source of contamination is fishing from the polluted waters, washing the catch with polluted coastal or harbor waters, and sorting the catch on contaminated sea beaches or contaminated onboard deck utensils, etc., and use of contaminated water for processing, and handling the material by workers who are carriers of salmonella, and improper personal hygiene, and entry of flies, rodent, and wall lizards in the processing hall. And symptoms, it will start within 12 to 24 hours of consuming of infected food. And usual symptoms are nausea, vomiting, abdominal pain, restless, diarrhea, etc. And it can cause typhoid and paratyphoid. Salmonella typhi causes the typhoid, and paratyphi causes paratyphoid. And this comes with the Listeria monocytogenes, and it is most foodborne pathogen, and outbreak of this disease is very rare because it is considered to be very serious due to high rate of mortality. And they are gram-positive and microaerophilic non-spore forming motile rods. Habitat, it is widely distributed in nature, and the variety of the animals can serve as a host for this organism. And it is found in the intestinal tract of domestic animals, birds, and human. About 1% of the human population is known to carry Listeria monocytogenes. And here one of the work which are studied over here, and we found that about 2.5 incidence of this Listeria in fish, which is collected from the landing center, is about 2.5%. And they can grow in the salt solutions with up to 14% of sodium chloride, and can survive in freezing temperature or even grow in fish at refrigerated temperatures. And it is called as, and it one of the main concern of the Listeria monocytogenes is that they can able to proliferate in the ready to eat food products where there is a lack of inactivation steps before consumption. And it was found that the fish skin is considered as one of the primary contamination source, and there is reports of Listeria. There is a incidence rate is very high. Range is reported, that is from 1.4% to 78.4%, and there's a lack of post-harvest practices are major point of Listeria monocytic contamination in fish and fish products. And the symptoms which is caused by the Listeria, it's a general name of disease caused by this organism. And Listeriosis is the disease which is caused by this organism. And the symptoms are include the meningitis, abortion, septicemia, and a number of other maladies, some of which may lead to death. And mortality is very high, about 30%, but in untreated cases, it may up to 70%. And control measures is the adequate sanitation and proper control measures to be followed. And coming to the, these are the around the last part of the presentation, whether they're different toxin detection methods or the presence of this toxin which are produced by this bacteria in the food is detected by usually detected by the four method. One is a bioassay method, in which uses the whole animal assay or cell culture assay, or immunological assay method, which is the ELISA test, or RPLA, that is a reverse passive lattice agglutination assay. And also you can use the mass spectrometry for the detection of this toxin, that is LCMS or MS, and MALDI-TOF or MS. And finally, we can detect the presence of genes of in the bacteria using the PCR by using the conventional or real-time PCR. That's all.
Participants, if you have any questions, please ask. Any questions? Or you can write down it on chat. I request Dr. Faisal, sir, please present the vote of thanks to ma'am.
Hello. Hello, thank you, ma'am. Thank you for nice elaborative presentation. If you are having any questions now, you please ask questions, because discussion is important in these 21 days program. If you are having question, you can write in chat box so that we may have elaborative discussion.